Hello and welcome. My name is Moshe Giladi. I joined Novocure 15 years ago, and for the past 10 years, I lead our internal preclinical research activities, as well as our research collaboration with many leading institutes worldwide. I feel fortunate to study TT field ingenious invention of Professor Yoram Palti and take part in expanding the knowledge of this novel electric field-based therapy. While there are multiple other modalities in medicine which utilize electric fields, TT fields are unique in their frequencies, intensities, and their mode of delivery. By tuning the frequency to several hundred kilohertz, we now have the ability to continuously generate an electric field within the cell. By doing so, we mainly affect processes which need to be carried in a very precise and orchestrated manner, and that have a detrimental effect on cell fate. Probably the best example is mitosis, a process where a single cell divides into two identical daughter cells, as you saw in the movie. From the very early days, we noticed that TT field disrupt key structural elements of mitosis, thus leading to mitotic cell death, or to the formation of compromised daughter cells. With that knowledge, we and others, as you will hear from Dr. Hagemann, started testing the combination of TT fields and antimitotic agents, discovering synergistic interaction in many of these cases. 3 of Novocure phase 3 studies in non-small cell lung cancer, ovarian cancer, and pancreatic cancer combine TT field with antimitotic agents. TT field antimitotic effect is only the starting point for multiple other downstream outcomes. Those cells which did not die during mitosis are compromised. They divide slowly, their ability to form colonies is reduced, and many of them will die shortly afterwards. Not only that, these compromised cells also demonstrate various forms of stress, such as induction of autophagy and ER stress. These are the key drivers for immunogenic cell death, the kind of cell death which prime the immune system to go after the dying tumor cells. Realizing that TT fields could kickstart the cancer immunity cycle, we started combining TT field with immune checkpoint inhibitors. Novocure Lunar phase 3 study in non-small cell lung cancer and our recent study with Merck test the combination of TT fields and immune checkpoint inhibitors. It doesn't end there, as TT field could prime the immune system in other ways, as you will learn later from Dr. David Tran. In recent years, multiple publications demonstrated that TT field interfere with DNA damage repair, which, like mitosis, need to be performed in a very accurate manner and have a detrimental effect on cell fate. Indeed, the combination of treatment which induce DNA damage, such as radiation therapy with TT field, or better yet, with irradiation and PARP inhibitors, proved to be highly synergistic in preclinical models. Novocure recent phase 3 study in GBM Trident and our studies in mesothelioma, pancreatic cancer, and gastric cancer combine TT field with modalities which induce DNA damage. Through the understanding of the underlying mechanism of action, we now have a better understanding of why some cells are more susceptible to the effect of TT fields. Based on this, we are exploring combination strategies to sensitize the less susceptible cells and enhance TT field efficacy. The story of TT fields continues to evolve all the time. In the past 2 years, we learned that TT field could affect cell membrane permeability and could also affect the blood-brain barrier integrity. Of this, you will hear more from Dr. Hagemann. Novocure's ability to make breakthroughs in some of the most aggressive types of solid tumors is the result of continuous investment in R&D and in the science of TT fields. We are developing preclinical research tools which are offered to our many excellent collaborators worldwide. In my years in Novocure, the number of our preclinical research scientists had multiplied by 20. This year marks our highest investment in R&D to date. We now have grant mechanism to support TT field research together with the American Association of Cancer Research, AACR. I strongly believe that in the upcoming years, TT field research will reveal many new outcomes of this therapy and will allow us to further improve treatment efficacy. I'm now going to turn it over to my colleague, Dr. Carsten Hagemann.
Thank you, Moshe. Dear ladies and gentlemen, for the first time, I heard about TT fields in 2014 at the EANO meeting, and honestly, this was very interesting because we just had published our data on synergistic effects of spindle checkpoint inhibition in combination with the spindle poison vincristine. The inhibited protein kinase, MPS1, is a key regulator of the mitotic spindle checkpoint. It detects aberrant spindle fiber attachment to the sister chromatids during mitosis, as they are caused by vincristine, regulates multiple protein complexes, and thereby leads to a mitotic cell cycle arrest. This provides time for the cell to repair the damage, so the cell might divide normally, even with initial spindle damage. Since the effects of TT fields on spindle fibers seem to be very similar to those of vincristine, we wondered whether a combination of spindle checkpoint inhibition and TT fields would synergistically augment the TT fields effects. We reproduced the known effect of TT fields at 200 kHz for our glioblastoma cell lines and treated these cells with TT fields in combination with MPS1 inhibition. While the single treatments led to a reduction in cell proliferation, The combination resulted in reduced cell numbers compared to the start of the experiment. This was due to increased cell death resulting from aberrant mitosis. The upper panel shows normal mitosis and on the right normal nuclei of the cells, while the lower panel highlights the combination treatment leading to mitotic catastrophe indicated by the highly aberrant shape of the nucleus. We published these data in 2018. At this time, a colleague approached me with a plan to investigate drug treatment of the blood-brain barrier to better treat glioblastoma. The blood-brain barrier protects the brain from harmful substances, including drugs transported by the blood, and is a special composition of the brain's blood vessels. The vessels are formed by astrocytes, pericytes, and endothelial cells. The latter are connected by tight junctions, which densely seal the vessels and are the major structure of the blood-brain barrier. They prevent that many effective drugs can reach the targets inside the brain. Since any new drugs must be used in conjunction with the standard therapy, we tested the endothelial cells with TT fields. We grew these cells as monolayer, stained the tight junctions at the cell-cell boundaries, shown in green, and treated them with different frequencies of TT fields. I did not expect to see any effect because TT fields had not been described to affect normal, non-malignant cells. Therefore, I was very surprised to see such major delocalization of the tight junction proteins when using TT fields at 100 kHz. Transendothelial electrical resistance was reduced, indicating opening of the blood-brain barrier and allowing blood-brain barrier impermeable molecules to pass through. Most striking was that these effects were reversible, so that the blood-brain barrier recovered within 72 hours after TT fields were switched off. The team of Dr. Ghilardi performed animal experiments for us. Mice received the contrast agent gadolinium, which does not pass an intact blood-brain barrier, and MRIs were taken before, directly after, and 96 hours after end of TT fields treatment. The contrast agent only passed the blood-brain barrier and accumulated inside the brain in case of TT fields application. 96 hours after end of TT fields treatment, the blood-brain barrier recovered, confirming our in vitro data. We even have first results that the TT fields-induced opening allows blood-brain barrier impermeable drugs to pass through for an effective treatment of glioblastoma. This research of the last blood-brain barrier opening through TT fields and its subsequent recovery could lead to possible clinical strategies for enhanced and more effective drug delivery for the treatment of brain tumors and other diseases of the CNS. Thank you for your attention.